and I'm here to take you through pharmacology revision questions and answer series. So this is the first series of revision questions, so I'll be posting as many revision questions and answers as possible. So apart from pharmacology revision questions and answers, I have also posted videos on biochemistry. So I have posted questions and answers in biochemistry, so how to easily remember answers in a given essay in biochemistry. So to get the whole of that work, search for the name Tom Rugenda Waswa. That is the name of the channel. If you get it, you will be there. So we begin. Using specific examples, distinguish between receptor and non-receptor mechanism of drug action. So that is the first question in the series. So using specific examples, distinguish between receptor and non-receptor mechanism of drug action. So to easily answer that question, you need to ask yourself several small questions. So you need first of all to ask yourself, what is a receptor? What is a receptor? Then another question that you need to ask yourself is, what are these mechanisms of drug receptor interactions? Then what are these specific drugs that interact with the receptors using given mechanisms? Then another question that you need to ask yourself is, which term is used to describe a drug that is able to bind to a receptor? Then you see, when a drug binds with a receptor, there are two possibilities. A drug may bring physiological effect or may not bring a physiological effect. So what is the name given to a drug that binds to a receptor and bring a physiological effect? Then what is the name of a drug? What is the name given to a drug that binds to a receptor but does not bring about physiological effect? So that is a very, very important part of your mind. That is during the revision of pharmacology questions, and also in biochemistry. That's the way you are supposed to ask yourself questions and then answer them correctly. So we begin. So what are receptors? So receptors refers to protein molecules that are either found on the surface of a cell or inside the cell of a given organism. So most of the drugs interact with the receptors either through simple physical means or through chemical means. So drugs that interact with receptors include insulin receptors, cholinereceptors, and also adrenergic receptors. Then endogenous neurotransmitters, corticoids, and also hormones produce their effect by binding with specific receptors. Then, so what are the drugs? So some drugs such as aluminum hydroxide and also magnesium trisilicate, they act through simple chemical means. So these are the drugs that are used for treating peptic ulcers. So they act by neutralizing gastric juice in the stomach. So another thing that you need to ask yourself is the ability of a drug to bind to a receptor. So what is it? So the ability of a drug to bind to a receptor is known as drug affinity. Then the ability of a drug to bind to a receptor and bring about physiological effect is known as drug efficacy. So you also need to know the difference between drug affinity and drug efficacy. Then what is the name given to a drug that binds to a receptor and brings about pharmacological effect? So the name given to such a drug is agonist drug. Agonist drug. So an agonist is able to bind to a receptor and bring about physiological effect. On the other hand, an antagonist refers to that drug that binds to a receptor but does not bring about physiological effect. So the other part of the question that I said that you must ask yourself to answer that question is, so what are the forces that bind a drug to a receptor? What is the name given to those forces? So the name given to forces that bind a drug to a receptor is known as bonds. So drugs drugs are bound to a receptor by what we call bonds. So examples of the bonds that bind a drug to a receptor include that is hydrogen bond, covalent bond, ionic bond. Then we also have van der Waals forces. So among the bonds, we need to know which ones are the strongest. So the strongest are covalent bonds. So the covalent bonds are always permanent, that is during drug receptor interaction. So drug receptor complex is normally permanent and brings about physiological effect or biological effect. So during the interaction of a drug with a receptor, the drug may form a drug receptor complex which can still be broken down or dissociated into a drug and a receptor. So it means that the other forces, apart other than covalent forces, the other forces are reversible. So the drug receptor complex is reversible, it can be dissociated. So that constant that involves the dissociation of a drug receptor complex is known as constant of dissociation. So during the binding of a drug and a receptor, that constant of interaction between drug and a receptor is known as constant of association. We are associating a drug and a receptor. Then if a drug receptor complex brings about biological effect, that is it is known as intrinsic activity of a given drug. So that is what you are supposed to know. So once again we go through, I've said that receptors refers to protein molecules that are found either on the surface or within the cell. So most of the drugs interact with receptors, while others act through simple chemical or physical means without interacting with the receptor. So such drugs include cholinesterase, insulin receptors, and adrenergic receptors. Then endogenous neurotransmitters or thyroid hormones and most of the drugs produce their effect by binding with specific receptors. Aluminium hydroxide and magnesium trisilicate, which are used in treatment of peptic ulcer disease, act by non-receptor mechanism by neutralizing gastric acid. I think I have mentioned this. Then we have drug affinity. Drug affinity. So this refers to the ability of a drug to bind to a receptor. Then we have drug efficacy. Efficacy refers to the efficiency of a drug. That's the ability of a drug to bind to a receptor and produce physiological effect. Then we have an agonist drug. An agonist drug, it has both affinity and efficacy. While antagonist drug has affinity but lacks efficacy. So an agonist drug has ability to stimulate a receptor and mimics endogenous transmitter. So antagonist drug, on the other hand, blocks a receptor. Forces that attract drugs to receptors are known as bonds. So that is the name given to forces that attract drug to receptors. They are known as bonds. So I gave out examples of those bonds. That is the hydrogen bond, the ionic bond, covalent bond, and van der Waals forces of attraction. Then I have said that the covalent bond is the strongest and the drug receptor complex is reversible. So looking at our equation, we have a drug interacting with a receptor to form a drug receptor complex. So that constant of interaction of a drug and a receptor is known as constant of association. I have denoted as K1. If a drug receptor complex can be broken back into a drug and a receptor, that is known as constant of dissociation. So the forces that bring about these are these other three bonds other than covalent bond. Then if a drug receptor complex brings about biological effect which is not reversible, that is known as intrinsic activity. So our constant of association is known as intrinsic activity. Very, very important. So we go to the second question. The second question says, describe drugs under the following subtopic. Number one is A, that is number one is site of action. Site of action. So a drug may act extracellularly, that is on the outside the cell, that is. So example of such drugs is plasma expanders and also osmotic diuretics. Then also a drug may act on the surface of a cell. Those are penicillins and catecholamines. They act on the surface of a cell. Then we have some drugs that act inside the cell. They act inside the cell, that is anti-cancer drugs and also steroid hormones. So we continue with the questions. The next point, that is dose-response relationship. So how are we going to describe a drug under dose-response relationship? Actually, the exact relationship between dose and response of an organism depends on the type, amount of a drug being administered, and also the type of an organism under study. So when we plot a graph of response against the logarithm of a dose, we come up with a sigmoid curve. So the minimum amount of a drug that elicits response in an organism is known as minimum dose, while the maximum amount of drug, that is the highest concentration of a drug after which further increase in concentration will not change the response, is known as maximum dose. So I want us to look at the graded dose effect. As the amount of a drug administered to a single subject increases, the pharmacological effect also increases up to a ceiling effect. Then under control dose effect, sensitive organisms will become highly sensitive. Organisms will respond to drugs very fast, while some of the organisms will be resistant and they will require higher concentration of a drug for them to respond. So what are the characteristics of a control dose effect curve? So these are the characteristics of a control dose effect curve. We have median lethal dose and median effective dose. So from the word lethal, median lethal dose. So this refers to the dose in milligrams per kilogram that is required to kill half of the population of a given organisms. Then we have median effective dose. From the word effective, we can say that this is a dose in milligrams per kilograms that is required, that is desired to produce a required effect. That is the desired effect or the physiological effect in 50% of the population of organisms. Then we have something else that you are also supposed to know is therapeutic index. So what is therapeutic index of a drug? So it's the approximate assessment of the safety of a drug. It's known as therapeutic index of a drug. So it is the median lethal dose divided by median effective dose. So we can write it as LD50 divided by ED50. So we go back. So this is what we are saying, that the exact relationship between the dose administered and the response depends on the organism under observation and the type of drug used. So when the graph of logarithm of a dose administered on x-axis and response on y-axis are drawn, a sigmoid curve is obtained. So the lowest concentration of a drug that elicits response is minimum dose, while the highest concentration after which further increase in concentration will not change is known as maximum dose. Then I have talked about graded dose effect and control dose effect. So graded dose effect, as the dose administered to a single subject or tissues increases, the pharmacological effect increases up to a ceiling point. Very, very important for you to know. That's up to a ceiling effect. So the concentration that is required to produce 50% of the maximum effect is termed as EC50 or ED50. So just know that effect is for EC or ED. Then we have control dose effect. So sensitive organisms or tissues give response to small doses of drugs, while some will be resistant and may require very large doses. So we go to characteristics of our control dose effect curve. As we've just discussed, we have median lethal dose and median effective dose. So from the word lethal, we can get the word lethal. Lethal, that is median lethal dose, we can get lethal. Lethal means to kill. So median lethal dose refers to the dose in milligrams per kilogram, which is expected to kill half of the population of the same species or strain. Then we have median effective dose. So median effective dose is also referred to as ED50. So this refers to the dose in milligrams per kilogram, which produces a desired response in 50% of the test population. Then we have also discussed about therapeutic index. So therapeutic index refers to the approximate assessment of the safety of a drug. So it is the median lethal dose divided by median effective dose. It's also called therapeutic window or safety of a drug. So therapeutic index is also known as therapeutic window or safety of a drug. So the higher the therapeutic index, the safer the drug. So a drug such as penicillin has got a very high therapeutic index. So we go to another discussion of a drug under structural activity relationship. So the activity of a drug is related to its structure. So why do you think we should know the structure of a drug? Number one is to find the competitive, that is to synthesize competitive antagonist. Number two is to synthesize compounds with less adverse effects and also specific actions. Then another point is it helps in understanding mechanism of actions of a drug. So in summary about structural activity relationship is that it helps us to come up with come up with competitive antagonist. Helps us to come up with competitive antagonist. Number two, it helps in the synthesis of new compounds with more specific and fewer adverse effects. So we come up with compounds with more specific and fewer adverse effects. So another importance of structural activity relationship is that it helps us in understanding the mechanism of a drug. So how do you recall that? You just remember ACM, that is antagonism. This one stands for antagonists. Then this one is the new compounds with the similar structure but fewer adverse effects. Then we understand the mechanism of action of drugs.